Good morning dear students of class 12 science. Welcome to biology class. We are dealing with the chapter biotechnology, principles and processes. We have done till the first step of process of recombinant DNA technology, that is isolation of genetic material. Try to recall that we are learning about the process of recombinant DNA technology. It's in the page number 590. Clear? So we have done the first step or the first process, that is isolation of genetic material, where we have to isolate or extract the DNA in its pure form, and this we have done by the help of different enzymes, where we have digested the macromolecules like RNA, proteins, which are present or which are mixed with the DNA molecules or the segment of DNA. Now, the second process of recombinant DNA technology is separation of isolated DNA fragments. Clear? So once we have taken out the DNA fragments or DNA from the plasmid vector DNA or from the host genomic DNA, then we have to isolate it. Okay, so this is mainly done by the help of the technique known as agarose gel electrophoresis. Clear? So separation of isolated DNA fragment is done by the help of the process known as agarose gel electrophoresis. So kindly write this step. Okay, so first one, remember that DNA fragments are negatively charged. Okay, due to the presence of phosphate groups, the DNA fragments are negatively charged, and these fragments of DNA can be separated by moving them towards the anode under electric field having a positive charge. And for this we require a medium or a matrix where DNA has to be separated, and that medium or the matrix which we are going to use is mainly the agarose, A-G-A-R-O-S-E, agarose. Okay, a natural polymer which is extracted from the seaweed. So first step is that the DNA fragments are negatively charged and hence can be separated by moving them toward the anode under an electrical field through a medium or a matrix such as agarose. So the charge of the DNA is negative, so it must be moved towards the positive charged anode, because positive charged anode will attract the negatively charged DNA fragment. Okay, so the DNA fragments will separate according to their size. Okay, the smaller fragments will move the farthest distance. Now when the DNA fragments will be separated according to the size, so the separated DNA fragments can be visualized after staining it with the help of ethidium bromide. Okay, the separated DNA fragments can be visualized after staining it with the help of a stain known as ethidium bromide. After that, it has to be exposed to the UV radiation. So a bright orange color will be available, and that is mainly a DNA. Clear? So now separate DNA fragments will be cut from the agarose gels, and they will extract it from the gel piece. Remember one more time, the separated DNA fragments will be cut out from the agarose gels and they will be extracted from the gel piece, and this process is known as elution. Remember the word elution. What is elution? It's mainly the process where the separated DNA fragments will be cut out from the agarose gel and will be extracted into the gel piece. Clear? And these purified DNA fragments will be utilized for constructing the recombinant DNA by joining them with the cloning vectors. So what we are doing in agarose gel electrophoresis, so this is the sample or the figure of the agarose gel electrophoresis. You can see the well over here. So from these wells we will put the sample that contains the fragment of DNA. The DNA is negatively charged, so by the help of the electrical field we will place an anode having positive charge. So negative charge DNA will be attracted towards the positive charge anode. Okay, so here we will look at a medium or a matrix to which the DNA fragments will move, and that medium or the matrix that we are going to use is the agarose. Okay, so under the influence of electrical field, the DNA fragments will start moving under the matrix or the medium. The smaller the size, it will move the farthest point. You can see over here the smallest fragment of DNA will move the farthest one, and the largest fragment will be close to the well. Okay, so by this way the fragments of DNA will be separated according to their size. Okay, now what we do, we are going to stain the DNA with the help of ethidium bromide, and after that we are going to expose it with the ultraviolet radiation, and this will give an orange band color of DNA. Now the DNA fragments will be separated or will be extracted or cut from the agarose gel. Okay, and this process is known as elution. So now the fragment of DNA which has been extracted from the gels can be used for the formation of recombinant DNA after ligating it with the vector DNA. So this is the overall process of of the agarose gel electrophoresis, which are mainly used to separate the fragment of DNA. So kindly copy that point. Okay, next one. Number third process is the cutting segments of isolated DNA at specific sites by restriction enzymes. So the isolated pure DNA will be incubated with the specific restriction endonuclease enzyme. Remember, these are the molecular seizure which are used to cut the DNA. Okay, so it will be incubated with the specific restriction endonuclease enzyme at optimal conditions, where DNA will be digested and will cut into smaller DNA fragment containing one or few genes. And after that, actually we will go for gel electrophoresis. Once we will extract the DNA, okay, from the host or the genomic one, we will treat it with restriction endonuclease enzymes, where it will be broken down into numerous fragments. Now those DNA fragments will be separated under the technique known as agarose gel electrophoresis. Clear? So after this, we will go with the fourth important step, that is amplification of gene of interest using PCR. Okay, so mainly what we have done, we have extracted the DNA, okay, in its pure form. Then we have applied it with restriction endonuclease enzyme, which will make a fragment of DNA. Now those fragments of DNA will be separated by the help of agarose gel electrophoresis. Okay, so each fragment of DNA can be utilized for the process of construction of recombinant DNA after its ligation with the vector DNA. But if you want to make a multiple copy of a DNA fragment, okay, of a single DNA fragment, then we have to go through the technique of PCR, polymerase chain reaction. It's in the page number 592. Okay, so polymerase chain reaction, or PCR, was developed by Kary Mullis in the year 1985, and for this he received the Nobel Prize in the year 1993. Okay, so polymerase chain reactions is the synthesis of multiple copies of the gene of interest. So here we can synthesize multiple copies of gene of interest from a single gene, or from a single DNA having the gene of interest. We can synthesize multiple copies of gene by the help of PCR. For example, from one piece of DNA having the gene of interest, we can create 100 copies of gene. Okay, so we can multiply, or we can create multiple number of gene of interest by using the technique called PCR. And here we will use the two sets of primer and the enzyme DNA polymerase. So what are primers? So primers are the small chemically synthesized oligonucleotides that have the complementary base pair to that of the DNA. Okay, so whatever you have got the DNA, the gene of interest, and whatever the base pairs are there, the similar type of complementary base pairs will be there in the primer. Okay, now the enzyme will extend the primer using the nucleotides and the genomic DNA template. So by continuous DNA replication, the DNA segment will be amplified up to 1 billion copies. So from a single copy, we can get around 1 billion copies by the technique of PCR. And for repeated amplifications, multiplications, we require a thermostable. Thermo means heat stable, which can reach high degree of temperature. So we were going to use a thermostable DNA polymerase enzyme, okay, in PCR, which can reach the high degree of temperature, while during the denaturation of DNA. When we used to denature the double-stranded DNA by applying the heat, we require such an enzyme that can resist such high degree of temperature. So we are going to use the thermostable DNA polymerase enzyme, which is mainly the Taq polymerase TAQ. Okay, Taq polymerase enzyme, which is obtained from the bacterium called Thermus aquaticus. Can you remember? We are going to use the enzyme Taq polymerase TAQ, Taq polymerase, which is obtained from the bacterium Thermus aquaticus, and the property of this enzyme is that it can resist high degree of temperature during the process of denaturation of double-stranded DNA. Okay. Now, the amplified fragments can be used to ligate with the vector DNA for the further cloning and for the construction of recombinant DNA. Okay, so now we are going to talk about the steps which are being used in the process of PCR for the creation or for the multiplication of the segment of the DNA having the gene of interest. So mainly there are three main steps in the process of PCR. Mainly first one is denaturation, second one is annealing of primer, and third one is extension of primer. So what is denaturation? So remember, my dear, that DNA is double-stranded molecule. Okay. So denaturation means the double-stranded DNA has to be converted into single-stranded DNA. So during denaturation, the double-stranded DNA will be denatured using high temperature to form the single-stranded DNA, which will act as the template DNA. Okay. So during denaturation, look at the figure. So during denaturation, this is the first step. During denaturation, what is going on? The double-stranded DNA. So you can see over here, this is the double-stranded DNA. So during denaturation, by applying heat, the hydrogen bonds, which are the hydrogen bonds between the complementary strands of DNA, so these bonds will be broken down, and as you can see, the construction of the single-stranded DNA. So this is called denaturation. So the hydrogen bond between the DNA will be broken down, and it will form a single-stranded DNA. This is the first step of PCR, that is denaturation. Okay. Second one is called annealing of primer. So two sets of primers are annealed or hybridized at low temperature using suitable enzymes based on the length and the sequence of primer. So you can see over here that two sets of primers are being hybridized or annealed with the template DNA. Remember that single strand of DNA will act as the template for the synthesis of new strand of DNA. Okay, so in each template we will require a primer which will continually lead to the formation of new strands of DNA. So therefore primers will be annealed to the template DNA strands. To two sets of primers will be annealed or will be hybridized to the DNA template. So this process is known as annealing. Clear? Okay, now the final step is extension. The primers are extended by adding nucleotides complementary to the template by the Taq DNA polymerase. So that means, so final step in extension what means that the primers has been annealed to the template DNA. Now by the help of the enzyme DNA polymerase, and mainly we are going to use the enzyme Taq polymerase. So this Taq polymerase will help in the replication or will help in the continuous synthesis of new strands of DNA. So the template was being annealed over here. Now you can see that the DNA has been, or the new strand of DNA has been synthesized. Okay, so this is called extension, where the primer has been extended and where the new copies of DNA has been created. So you can see over here that from one copy of DNA, so from one copy of DNA, two new DNA has been synthesized. Again we will continue the process. Okay, so again from here, from this strand of DNA, again two DNA strands will be synthesized. Again from here two DNA strands will be synthesized. So from 1 to 2, 2 to 4, 4 to 8. By this way the number of DNA having gene of interest can be multiplied by the help of PCR. Okay, so there are three stages of PCR: denaturation, annealing, and extension. And by the help of the process, the DNA having gene of interest can be amplified, can be multiplied 1 billion times. Clear? So today we have mainly learned about the separation of DNA fragments by the help of agarose gel electrophoresis. Then we have learned about the cutting down of the DNA segments by the help of the enzyme restriction endonuclease enzyme. Then finally we have learned the process of PCR, polymerase chain reaction, developed by Kary Mullis, where the DNA having the gene of interest can be amplified up to 1 billion times. Okay, and here we require the help of a thermostable bacteria enzyme that's called Taq polymerase. Okay, thermostable DNA polymerase enzyme which can reach high degree of temperature, and that enzyme is Taq polymerase obtained from the bacterium Thermus aquaticus. And you can usually ask yourself also, like, why we used to require the enzyme that must be thermostable. Okay, because during denaturation of DNA, in the first step we are applying our heat, or we are usually applying the temperature up to 90 to 100 degrees Celsius to mainly break down the hydrogen bond between the DNA strands. Okay, so in order for the enzyme to become functional, the enzyme must reach this high degree of temperatures. So we require a thermostable enzyme, that is Taq polymerase, which can reach this high degree of temperature up to 90 to 100 degrees Celsius. This Taq polymerase can reach its temperature up to this. Okay, so this enzyme is being employed for the technique of PCR, and this enzyme is responsible for the extension of primers, leading to the formation of new strands of DNA. Clear? So we'll continue with the same topic, that is process of recombinant technology, in the next class also. So we have done till the fourth step, that is amplification of gene of interest using PCR. So the next remaining steps we will do it in the next class. Okay, so this much for today. Thank you.